Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Revisiting and Rebuilding Clinical Development of Drugs for Antimicrobial Resistance, Lessons Learned During the COVID Pandemic. My name is Sydney, and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved, so please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank MedPace, GARDP, and IHMA who developed the content for this presentation. MedPace is a scientifically driven, global, full-service clinical contract research organization providing phase 1 through 4 clinical development services to the biotechnology, pharmaceutical, and medical device industries. MedPace's mission is to accelerate the global development of safe and effective medical therapeutics through its high science and disciplined operating approach that leverages local regulatory and deep therapeutic expertise across all major areas. Brian Murphy is Vice President in the Medical Department at MedPace and an infectious diseases physician. The Global Antibiotic Research and Development Partnership, GARDP, is a not-for-profit organization developing new treatments for drug-resistant infections that pose the greatest threat to health. GARDP works with partners to ensure sustainable access to treatments, promoting responsible use and affordability to all in need. Jennifer Schneider is Senior Advisor in External Affairs at GARDP. IHMA provides a full suite of technical capabilities and domain expertise in all phases of antimicrobial drug development, drug discovery, clinical development, regulatory approval, and commercialization. Daniel Somm is Vice President of Global Microbiology Services and Chief Scientific Officer at IHMA. And now, without further ado, I'd like to hand the presentation over to our first speaker, Brian. You may begin when ready.
Thank you, Sydney, and hi, everyone. Thanks so much for joining us today. I'm super excited to have the opportunity really to join this group and discuss antimicrobial resistance. And I think, if we are going to approach this problem successfully, it has to be in a multidisciplinary way. And by bringing various groups together, stakeholders, such as we have on the phone today from MedPace, IHMA, and GARDP, as well as the companies that you represent to discuss this topic, I truly believe we can move the AMR programs along. I know this audience is well aware, even before the COVID-19 pandemic began, that antimicrobial resistance was among some of the top priorities for global public health. But the current pandemic has given us all really, I believe, a deeper appreciation of the urgency in addressing some of the obstacles to AMR drug development. And we'll be talking about those today. So if we go into the next slide, we'll start through this. I think some of the shortcomings that we've all noticed over the last year in our medical infrastructure, public health, economic infrastructures have really been highlighted by COVID-19. But to find the silver lining, I think that the pandemic has drawn much attention to some of the threats that have been posed by emerging infections, and again, reminded us all of the importance of investing in public health. So in this talk today, we're going to explore some of the potential opportunities that are emerging for antimicrobial resistant drug development from the COVID-19 pandemic and how we can realize the potential of these drugs. We'll first discuss some of the changes that we're seeing during the pandemic, specifically regarding antimicrobial usage and infection control practices, and then discuss how those changes can potentially be leveraged and how the momentum of the pandemic, or at least the perspective from the pandemic, can be used for the research and development of new and effective antibiotics. The next slide will next discuss how we can reevaluate just how antimicrobial resistance has changed throughout the pandemic, and the importance of to continue to monitor resistance actively as this pandemic continues to evolve. I know for many of us in infectious diseases, the potential for the exacerbation of the problem of antimicrobial resistance during the pandemic has been something that we've all really been watching closely. I think the audience that's on the phone in this group really knows this, unfortunately, all too well. But many doctors in many of the regions that have been hit hard by this pandemic are desperate to manage their patients in an effort to save their patients' lives. Many of them are really using large amounts of antibiotics to treat these patients, and that's at the risk of developing resistance. Some of the initial data from the earliest time in the pandemic coming out of China suggested that perhaps 1 in 7 patients that were hospitalized with COVID-19 acquired secondary bacterial infections, and it's possibly because of that observation that drove some of the initial responders to increase the use of empiric antibiotics for patients coming in where they weren't sure if it was COVID-19 or a bacterial pneumonia. And now subsequent data from other regions have of course suggested that maybe bacterial and co-infection COVID-19 occurs less frequently, but we'll talk more about that. Clearly, COVID-19 has highlighted the fact that data collection for infectious disease needs to be strengthened and broadened, and needs to become more active in integrating local and regional surveillance systems to account for some of this heterogeneity in specific hospitals' antibiotic prescribing behaviors and resistance trends. And if we go to the next slide, we'll finally wrap the webinar up by looking at some of the potential paths forward. The pandemic has definitely highlighted the fragility, I believe, of the antibiotic pipeline and the gaps between our currently available antibiotic arsenal and the need for pipeline of new antimicrobials that are not only adequate to cover the current risk, but sustained enough to be able to provide for future risk and potential future pandemics. So we desperately, of course, I think as everyone is painfully aware, need improved mechanisms for pooled funding, new partnerships, and investments of people willing to pursue these opportunities, human capital, if you will, in order to meet really the magnitude of what is the antimicrobial resistance threat. So if we go to the next slide, then let's look at some of the numbers associated with antimicrobial resistance. And of course, it's hard to have a discussion about this topic and not really talk about the use, the misuse, or even the overuse, perhaps, of antibiotics. Prior to the emergence of COVID-19, it was approximated over 250 million antibiotic prescriptions were given out annually, with about a fifth or so of those thought to be really unnecessary. And I think the pandemic has really invigorated or reinvigorated the conversation around antimicrobial stewardship and some of these numbers. During the first year of the pandemic, the use of antibiotics went through multiple stages. Initially, perhaps there were lower usage in regions and in times where patients were afraid to go to clinics or hospitals, and that transitioned into really increased use of outpatient antibiotics. We didn't have any drugs with approval or emergency use authorization for COVID-19. There was a lot of information and misinformation about what could be used. We saw prescriptions for antibiotics such as the azithromycin, amoxicillin clavulanate, for instance, really being consumed at high rates during those early parts of the pandemic. And then that shifted now to seeing the surge that was coming into our ICUs and hospitals, where we really saw a significant increase in broad spectrum antibiotic use in regions. And again, that was kind of lining up with some of the surges that we would see with hospitalizations. Some have reported that between 70 to 90% of hospitalized patients with COVID-19 receive antibiotics during the first 48 hours of their hospitalization. Data from Pew researchers found that about 52% of the overall COVID-19 patients were given at least one antibiotic, but really only 20% or so of those patients were eventually diagnosed with a suspected or confirmed bacterial infection. In the U.S., we report that there's about 3 million or so antibiotic-resistant infections by superbugs each year in the U.S. alone, and nearly 700,000 deaths worldwide due to drug-resistant diseases. Projecting that out farther, there's expected to be around 10 million or so potential deaths worldwide by 2050. We'll talk more about how that it's changed during COVID on some next slides, but to put that a bit more in the perspective, the total death rate for the first year of the pandemic was almost 2 million globally. If we look at the economic side of things, there's over $8.8 billion spent on antibiotics yearly. It's an estimated additional impact of around $5.7 billion or so to our economies just from 9 of the 18 organisms that are identified to be AMR threats. If that current trend continues, some of the European economic models project that the global cost of AMR would be around $5.8 trillion US dollars by 2050 if that current trends continue. Bringing this back to the global economy during the first year of the pandemic, the International Monetary Fund released data and showed that COVID's impact in the first year of the pandemic was around $3.9 trillion in lost economic output. So you can put these numbers in perspective with AMR and the pandemic. So these numbers are clearly impressive, but I think it's important to note that when we talk about AMR and COVID numbers, those are really often driven by countries where there's a large number of highly vulnerable people, such as India. And many of these numbers were developed even before the most recent COVID-19 surges occurred there. So clearly, we need to continue to monitor AMR numbers as the pandemic continues in this now second year, unfortunately. Go to the next slide, please. So let me take a little sidestep and speak just a moment about the potential synergy with the SARS-CoV-2 virus and bacteria. It's well known that many viral infections can promote the production of various bacterial infections, and definitely in respiratory infections by impairing immune responses and enhancing the destruction of cells and tissue. And of course, with SARS-CoV-2, a very intensive cytokine response is there. It's been well described. But our knowledge as to the exact degree that COVID-19 is complicated or made worse by bacterial or fungal superinfections, and how it does so is really still evolving. If we look back during the 1918 flu pandemic and some subsequent flu pandemics, Streptococcus pneumonia and other typically gram-positive bacterial superinfections were common causes of mortality and morbidity. And some recent studies have suggested that even fungal infections and aspergillosis are also under-recognized but really important complications in patients with severe influenza cases. Part of the reason for this association can be explained in how the respiratory viral infections alter the innate immune function in these tissues and promote bacterial growth. The macrophages, for instance, are easily overwhelmed or quickly overwhelmed by an increased burden of dead cells, apoptotic cells, and become limited even in their capacity to phagocytose other bacteria by some of the cytokines that are present. Antigen-presenting cells have diminished ability to present antigens following a viral infections, and even epithelial cells are altered, and in doing so, they're not able to clear the infection. Their mucociliary clearance is decreased, which can lead to increased thickening of the mucus and respiratory secretions. Adding on another factor, all of these processes can really change the respiratory tract microbiome, which is obviously an intense area of research for many of our companies and researchers. And altering that can in turn undermine immune defenses even more against these pathogens and create an environment that's favorable for various bacteria or even fungi. So despite some preliminary evidence, it's still not clear whether those same synergies that we've seen in influenza exist between SARS-CoV-2 and specific bacterial infections. In the setting of SARS-CoV-2, obviously the pulmonary alveoli do become filled with fluid and in some cases purulent material or pus, which does create a nutritive environment for organisms such as Pseudomonas or Staph aureus. Some of the initial data from China suggested that secondary bacterial infections were observed in about 15% of the COVID patients, but in 50% of all their deaths. That observation is really consistent with reviews of autopsy series and postmortem cultures from patients from the 1918 flu pandemic and subsequent flu patients, revealing that most victims had concurrent bacterial pneumonia. But unlike the gram-positive organisms that were observed in patients co-infected with influenza, some preliminary findings on studies from this pandemic focusing on co-infections in COVID-19 show that gram-negative organisms predominate, and these are especially Klebsiella, Acinetobacter, Pseudomonas H. flu has been described, and even some more recent studies coming out of India continue to demonstrate that gram-negative organisms are a big player, especially, again, Klebsiella and Acinetobacter, with really high rates of resistance to some of the higher generational antibiotics from those studies. Data have also emerged from other regions later in the pandemic, later than those initial observations from China, that have shown somewhat of perhaps a lower number of overall patients with COVID-19 with secondary bacterial infections, but the reports vary between 4 and 8%, definitely lower than what we initially heard reporting out of China. Still, the mortality rates exceeding 50% or so for those who do develop secondary bacterial infections. So still, if you look in the most severe and critical COVID patients, the number of patients with a secondary bacterial infections are reported to be quite high. Around 54% or so of ICU COVID patients have a known or suspected secondary bacterial infection. Let's go to the next slide, please. So if we look at the epidemiology of the development of antimicrobial resistance during COVID, we can see some interesting trends. And unfortunately, it's like many other things. It's a bit of a tale of the haves and the have-nots. I think it's fairly intuitive, but the data do support the fact that in environments where personnel and PPE were insufficient during the pandemic, hygienic conditions deteriorated. They got worse, of course, and as well, and then the rates of resistance increased. There's a study by Thierry and team in Italy during a COVID surge, and they showed that the incidence of carbapenem-resistant Enterobacterialis acquisition in the ICU went from 6.7% to over 50% during that short period of time. And they really attributed this to a lack of PPE and a lack of experienced healthcare professionals that were associated with this increased risk of spreading carbapenem-resistant organisms, in particular Klebsiella pneumonia in those ICUs. They also mentioned that the high intensity of care and really even the positioning of intubated patients using more of a prone position required a lot of hands on those patients and additional inexperienced healthcare workers to come and manage those patients for extended periods of time. And that's part of the reason that drove the numbers. All of this high demand for care in the ICUs then really caused them to pull in more healthcare workers, perhaps with little to no ICU experience, and created a suboptimal experience to nurse-patient ratio. That trend's been described in a lot of other studies. One coming out of the U.S. showed that carbapenem-resistant Acinetobacter cases also increased when COVID surged in their units. In that study, they attributed the increase to corresponding shortages, not only in personnel and PPE, but also shortages in medical equipment that they were having to reuse very quickly with perhaps not adequate cleaning. So really, they in that publication, the changes in existing infection control measures, part of it in an effort to conserve PPE and resources, and pausing routine audits of infection control compliance was really to part of the issue. Other studies looking more around some of the molecular causes of this, looking at clonal spread of pathogens associated with ventilator-associated pneumonia, including escape pathogens, demonstrated that the emergence of these bacterial infections secondary to COVID could be closely related to poor clinical practice by health personnel and some inadequately clean medical equipment. So no surprises there when you see that. But on the flip side of this, infection control and prevention measures have really been used on a greater scale in areas where resources were not limited. And when they have been fully implemented and widely used, of course, they work well, as we know. So increased uses of face masks, gowns, gloves, alcohol for hand hygiene, some of the restricted visitation policies really increased in many of these regions in 2020 as compared to prior years. Here's a study from Taiwan and from sites where they experienced no real shortage of medical resources and demonstrated that the overall incidence of hospital-acquired infections did not differ during the pandemic from the pre-pandemic period. And in fact, nosocomial urinary tract infections significantly decreased. In the same study, we reported the incidence of multidrug-resistant organisms were lower overall in 2020 than the pre-pandemic baseline, especially noting that carbapenem-resistant Acinetobacter and vancomycin-resistant Enterococcus were significantly lower during the first year of the pandemic than again that same time in the preceding year. There was no change in carbapenem usage compared to the pre-pandemic baseline in that study. but they did mention that glycopeptide usage had increased. Here's another study from a health system in Singapore where they report a significant reduction in central line-associated bloodstream infection rates during the pandemic period. And this one was quite interesting, and they note that their prior experience in dealing with SARS back in 2003, the severe acute respiratory syndrome, another coronavirus, of course, in 2003, led to the early adoption of enhanced infection prevention strategies and placed them kind of farther ahead than other institutions, perhaps. But in contrast, and to be fair, there were other hospitals in Singapore around that same time that reported that the pandemic negatively impacted their infection prevention practices and observed the opposite trend with some increasing bloodstream infection rates. This last one is a study from, again, in Italy, and where we saw some of those sites had resource limitations, other ones did not have such limitations, and reported a significant overall reduction in the incidence of total multi-drug resistant infections during the pandemic. They did report a higher incidence of multi-drug resistant infections in some departments. In many places, as you're aware, that there will be departments or whole hospitals that are dedicated to managing COVID-19 patients. So in those COVID-19 departments, they did have higher incidence of multi-drug resistance than they did in non-COVID departments, 29% versus 19% in this study. The pathogens, the MDR pathogens that were isolated most were, no surprises, MRSA and ESBL, Klebsiella pneumonia. They did report a significant decrease in ESBL producing Enterobacteriaceae compared to some of their pre-pandemic samples. Unfortunately, this site did have a high rates of, this system had a high rate of Acinetobacter, and that was the main pathogen in these COVID-19 patients, and definitely higher than it was pre-pandemic, almost 10% versus around 3% from the pre-pandemic. Just the other side of this, no change in Clostridium difficile in these patients with COVID-19, and those that were negative in the different departments. So just a quick introduction then, snapshot to where we are. I'm going to shift gears now a bit, hand it over to Dan at IHMA to look at some of the active surveillance that's going on with his group. So Dan?
Thanks, Brian. So what I want to talk with you about today is I'm going to give you a little bit of an overview of what's going on with antimicrobial resistance surveillance in general as a background, and then finish up with talking about, so what has COVID meant for these efforts that have been going on. Just as a reminder on this slide, there's a lot of different surveillance going on to monitor changing and emerging resistance patterns among our clinically relevant bacteria. It gives us a window into the world a little bit about what could be going on with the emergence of resistance. It's important for older as well as new approved agents. We really want to know if new agents, as they're released into the environment for clinical use, is resistance emerging, and if so, is it transferable? Will it explode? How do we keep an eye on it? And also, what are some of our oldie but goodies doing? The carbapenems have been around for quite a while, but they're very, very much a mainstay of our therapy. So we need to keep track of drugs that have been around for a while as well. The data that we get out of here, which we will discuss in a few slides ahead, can help to forecast some of our unmet medical needs. What kind of resistance or multi-drug resistance patterns are emerging, which would indicate that we need to start working on strategies quickly, as it does take 8 to 10 or more years to get a drug from discovery through to the regulatory filings. Optimally, when we do surveillance, they need to be conducted in a robust, continuous, and uninterrupted manner. And with funding in the antimicrobial world being as a challenge as it is, some of these surveillance networks or initiatives come and they go, and the interrupted manner that results in can lead to difficulties in tracking trends. And also, as we've learned more than anything else from this COVID pandemic, this antimicrobial resistance as well is a global challenge. It's not just any particular country's problem, And that's why antimicrobial surveillance needs to take on a global representation. Now, in the next slide, I've outlined some surveillance programs that perhaps many of you are familiar with. They come out of the government sector, whether it's the WHO or the European CSER network. There's a network of the CDC, the National Antimicrobial Resistance Monitoring System for Antibacterial, or NARMS, and China itself has a couple of networks where they're doing surveillance, and they all have they have variations in terms of their size, their scope, their target organisms, etc. But there are these government-based surveillance initiatives that are out there. But I wanted to, if we can go to the next slide, I want to really focus on two major surveillance initiatives that IHMA has been helping run for the last several years, and these are industry-sponsored surveillance networks. One is the Pfizer Atlas program, and the other is Merck's SMART program. I've provided websites, and I'll refer to those websites in a few minutes. There is no surveillance program, private or public, that fits all sizes. We need data from as many different corners of the world as we can get it. So I just wanted to present a perspective on these industry-sponsored ones because they are rather robust. They were established in the early 2000s and have been run annually since then. So we're coming up on over 20 years worth of data. They are global, with more than 55 countries involved. They do focus on the most common encountered clinically relevant gram-positive and gram-negative species, primarily those that are associated with hospital-based infections these days. We also monitor them based on for their body sites or site of infection, whether it's UTI, wound, blood, or respiratory tract. And also there's a good bit of demographic data, patient, gender, age, etc., that's captured with this data for trending along those lines. The surveillance is profiled against a wide variety of critical antimicrobials. We track new agents from pre-NDA stage to the end of a patient patent life and beyond, as well as some of those oldies and goodies that I referred to earlier that maintain a critical point of our medical care for patients, like the aminoglycosides and fluoroquinolones, etc. And many of the bacterial isolates that are collected, and I'll go through the mechanism of this surveillance in a few slides. They are also molecularly characterized so we can have a better understanding of what kind of mechanisms are emerging out there. Are they on transferable genetic elements, etc., which could be more problematic than just mutants that arise and die back. So if we go to the next slide, please, the output from these two large programs is really through two primary mechanisms. One is that over the years, scientific presentations at international and local conferences, vis-a-vis abstracts, posters, slide presentations, and publications in peer-reviewed journals have contributed greatly to getting this information that comes out of the surveillance networks out into the greater medical and scientific community, as hundreds of these communications have been delivered over the years and are continue to be delivered every year. In addition, I mentioned these websites before, and I encourage you to go visit them. They're both, the Atlas and the SMART are interactive websites, so you can see what's going on locally with the organisms of your interest and other kinds of parameters that you may want to measure antimicrobial effectiveness with. And the data is quite recent. It's within it would be within 1 year since the data was collected. It shows up on these websites. So again, this is another great way to view what these surveillance initiatives are telling us. So if we go to the next slide, I just wanted to talk to you about how the surveillance is set up and how it's run, because it really pertains to subsequent slide that I want to talk about, looking at what's COVID doing to these efforts. It all begins with the medical institutions that are invited to participate, and that's based on geographic distribution. Where in the world do we need to look? And those medical institutions, if they agree to participate, then we work with them to save, process, and ship the requisite isolates based on body site, et cetera, to our central laboratory in the US at IHMA. Once we receive them, we reconfirm the identifications, we profile the susceptibility of the organisms to a large number of clinically relevant drugs, and that this is all done by CLSI standard methodology, most commonly by broth microdilution. And then there's a subset of those isolates depending on their phenotypic profile that are molecularly characterized. So we, again, as I mentioned earlier, have a better idea of what might be the underlying emerging mechanisms. And then the organisms are stored, and that seems trivial, but I'll tell you why that's important when we get to the next slide. And then finally, we house all this data in our database so that it can be maintained and used for the output analysis that I mentioned on the first slide, either for publications, ad hoc analyses, or to support the interactive websites that were mentioned previously. So the medical institutions are the lifeblood of this, so just keep that in mind. Now, getting back to what I said, we just store organisms, that it really relates to collateral benefits of surveillance. The primary benefit being the data and the information we glean from our data, but also these bacterial isolates that are collected as part of the surveillance programs can be made available and are made available to industry and others to support the discovery and development of new antimicrobial strategies, whether those strategies be small molecules, biologics, phage therapy, etc. We've also, these isolates can serve as a resource to support other people's initiatives, such as the CDC and FDA and the Resistant Isolate Bank. Also, we use the isolates can be shared with diagnostic companies to help them develop rapid diagnostics for the detection and characterization of resistant organisms. So this organism bank that's built year after year can really use very current resistance patterns and known well-characterized organisms to help springboard other research into new anti-infectives and diagnostics. And furthermore, in the last bullet, the data that we can use can identify clinical site selection and inform feasibility of various studies and designs, especially if sponsors of the drug are looking to get an indication for carbapenem-resistant Acinetobacter or ESBL producing E. coli. The resistance patterns that are seen at some of these sites can be used to maybe perhaps use those sites as primary candidates for placing a clinical trial as well. Then the next slide. So what has been the impact of COVID on surveillance? Well, the primary one is under the first bullet point, because the medical centers, as I mentioned, is the lifeblood of surveillance. And the pandemic has greatly impacted the bandwidth at our sites in the surveillance networks that we have. And this has recalled a number of sites, not a tremendous number, but a number of sites have had to withdraw this past year in 2020 and not participate. We fully anticipate they can come back in 2021, but they just did not have the bandwidth as they were dealing with the obvious problem of COVID. And then the sites that did participate, there has been a reduction in productivity because, as I mentioned earlier, each site, for the robustness of the surveillance, is we sign them up to give us X number of strains from Y number of body sites, et cetera, et cetera. And again, just because of their bandwidth challenges, not all of them that did stay in the program were able to meet those numbers. So we'll probably have some weak pockets, if you will, this past year from the 2020 initiative. What about the potential impact of COVID on antimicrobial resistance patterns? This is, in terms of processing our 2020 data, which we're in the midst of now, it is too early to tell, but so far we've seen no notable patterns on a global scale of any sort of resistance emerging. I think that some of the things that Brian referred to were important notes from certain institutions in certain countries, but it just, at least in our surveillance program, we've not seen any pattern it's obvious to us that the resistance has increased or things have changed. But when we're done processing the data, we might have a better feel for whether or not that still holds true. We continue to fight the good fight with our network. We're very hopeful that they all very much understand the importance of global surveillance, and they are anxious to participate with us. And we are anticipating that they will come back on board as we're enrolling in 2021 here, and hopefully as the pandemic starts to wane. On the last note here, hopefully the COVID has heightened the public awareness of infectious diseases, and we don't all live in bubbles, and it certainly is true for COVID and how it spread throughout the globe. And I think we need to use this as an enhancement for awareness of antimicrobial drug resistance as well, because this knows no boundaries either. So the analogy with COVID is very clear and straightforward, in my opinion, because they're both global public health threats. And with that slide, I think I will turn it back over to Brian.
Yeah, thanks, Dan. Dan, thanks. And I think as you mentioned, there are maybe regional reports or even local hospital reports, but it's still too early perhaps to tell exactly how COVID has impacted AMR and some of these large active surveillance programs like you run. I think you called them weak pockets in the fact that there are some perhaps holes in the data from 2020, and the fact that some sites, just because of resource limitations, weren't able to fully participate. I think all of that really underscores the importance of pairing global surveillance programs like our run at IHMA and others with specific local hospital antibiograms and susceptibility reports if you're getting ready to run a study as a company in an antimicrobial resistant indication. Many of our companies are still looking to develop their antibacterial programs in indications that are very traditional: urinary tract infection, nosocomial pneumonias, someone looking for pathogen-specific indications, around highly resistant gram-negatives, in particular Pseudomonas acinetobacter, of course, CREs, some in gram-positives. But we need to really know, and those companies need to know exactly what is the antimicrobial resistance pattern in specific hospitals that they may be considering for specific sources. What is the susceptibility profile of an organism from urinary isolates for UTI or respiratory samples for HAP and VAP? So just to describe briefly kind of how we at MedPace have kind of integrate data coming from you and global surveillance programs. We've worked to pair those again with specific hospital information, and do that, as you mentioned, first of all, during our feasibility process, to triage and prioritize regions that we think will contribute to data that will meet the objectives of the company, will inform the key study design decisions and help make decisions on comparators. Understanding that this pandemic is still ongoing and that resources may continue to be impacted during the pandemic and constraints at different rates at different times at different places, and these resistance patterns may change really abruptly, even sometimes during the course of a study. So we need to be very careful with that. I will say one other approach that we've taken with these data and bringing in collaborations with other pharmacology and PK/PD modeling groups. We have on a number of occasions for studies been able to pair the real-world data from these surveillance networks to our feasibility process to develop real-world epidemiology studies and even develop in silico virtual trials to help our sponsors select the right antibiotic combination, dose, duration of treatment, and do a dry run of the study, if you will. And I think in a world dealing with a viral pandemic like COVID-19 and still unsure of what's going on with antimicrobial resistance, it is something to consider these kind of in silico or real world epidemiology kind of dry run studies before you actually maybe venture into a full randomized clinical trial. Sometimes even we've been able to use biomarkers that have been collected in studies or other studies and through various statistical methods identify groups or subgroups of populations that may respond to the drug differently, which of course is critical in developing these study designs and anticipating even later stage designs. And I mentioned during the course of the study, no one wants the surprise that as you're running a study in a particular resistant pathogen, that susceptibility has changed in due course over the course of that study, and now that pathogen is not there, or there's a new a resistance that's emerging right under your feet. So it's clear you have to stay on top of this in real time and keep abreast of the situation on a local way, as well as pairing it with the global surveillance. Okay, so for the last step on this, we'll look more at the pipeline of the antibiotics. If you go to the next slide, please. The pipeline of antibiotics and look at some of the partnerships and coalitions that are going to be useful to move these programs forward. If you look to the next slide, I think the pandemic has really, the COVID-19 pandemic at least, has really emerged at a crucial time for antimicrobial development. Last year, the number of new experimental drugs and vaccines targeting COVID-19 or SARS-CoV-2 directly went from 0 to over 1,200. During that same time, Pew Charitable Trust reported there were only 15 new phase 1 antibacterial projects in AMR. So obviously this group and people on the phone, we've all been discussing for some time how the pipeline for antibiotics is really inadequate to meet the AMR need. And with even the public and private investments to support development, this 2020 report that you're seeing here from the Pew Charitable Trust shows that the pipeline has really been fairly static, with only 14 antibiotics approved by various regulatory agencies since 2014. As of December 2020, there were approximately 43 new antibiotics that were geared toward treating serious bacterial infections in their clinical development. That's compared to 38 in 2014, so fairly flat across the board in terms of new antibiotics. Of those 43, there were 13 that were in phase 3, and historically there's only been about 60% or so of those drugs that enter phase 3 that eventually will be approved. Just some quick other interesting observations on these. 19 of the antibiotics that are currently in development have the potential to treat gram-negative escape. 15 of the antibiotics are active or could be active against carbapenem-resistant or ESBL Enterobacteriaceae, Acinetobacter, or Pseudomonas, and only about a quarter in the pipeline represent new drugs, novel drugs, or a new mechanism of action. A lot of them, almost half of the novel products that we just mentioned, are actually development for C. diff. I don't think there's any surprise if I told you, but of the 38 or so companies that have antibiotics in clinical development, only 2 rank among the top 50 pharmaceutical companies by sales, and over 95% of the products in development today are being studied by small companies instead of the large pharmaceutical firms that really once dominated the field. It's not really surprising, I think, to any of us, given the fact that 5 of these companies that brought new agents to the market in the past 3 years have already declared bankruptcy or completely gone out of infectious disease. So the precedent is not great there for large pharmaceutical companies to get back in, at least immediately, even though there are some promising trends. In that same time, these same pharmaceutical companies, they obviously have the capacity for infectious disease and can do it, even though some may be neglecting AMR organisms. They did participate in the surge of research that I mentioned around COVID-19 that was heavily represented by large pharma. Most of that was in vaccine development, of course. And just one more note on that, of the companies that are developing, more than 70% of the antibiotics in development are from companies that have no other products on the market and are not generating any revenue. So the idea of partnerships is critical with this. Dan mentioned old drugs coming back and using old antibiotics. Just as we relearned from COVID, I think that will continue to be a critical place in this pipeline. And these older antibiotics do have a crucial role in every day. About 80% of the recently approved antibiotics, in fact, were derivatives of the existing antibiotic classes. You could argue those already have well-established drug resistance as well, but most of the variations of the antibiotic drug classes that we've seen were from classes discovered back in the 1980s. If we go to the next slide, Dan, do you want to comment just a bit more on some of the antibacterials in preclinical development here?
Yeah, just very, very briefly. What this slide shows is where some of the dynamics are going here. On the left-hand slide, the first of this is the percent effort in the first 3 bars there of small molecule, if you will, and that constitutes about 60% of the effort out there in preclinical projects that are small molecule, which means upwards of 40% are from the middle bar, antibodies and vaccines, and to the right, which are much more scientifically driven, esoteric new strategies from phage therapy, et cetera, that are being looked at. So there's a lot of effort going into non-small molecule strategies to fight drug-resistant bacteria. The challenges with them is they are scientifically complex. They're difficult to translate into clinical trials. The FDA is not necessarily familiar with everybody's learning how you even make these things a product. So there's a lot of effort, and we just wanted to point out there's a lot of effort in the non-small molecule, but there's a long way to go to make those successful and become a common path. And that's just basically what we wanted to point out on this slide, that there's other strategies besides small molecule.
And if you go to the next slide, I think looking at those non-small molecules, even beyond and putting them into the clinic, this is from a report from the 2020 WHO pipeline report. And for the first time in this, they show a nice comprehensive overview of non-traditional antibacterials or the non-small molecules. It highlighted 27 non-traditional antibacterial agents in the pipeline. And you've already heard about some of these: bacteriophages, live therapeutics, monoclonal antibodies, vaccines, immunotherapies, virulence inhibitors, microbiome-related agents are some of them, just to name a few. And I think it's just as I mentioned, we saw a dearth of large pharmaceutical companies pursuing more traditional antibiotics. A third really of the non-traditional antibacterials in development are being pursued by the top 50 pharmaceutical companies by sales revenue. And again, most of those are around vaccines. I think it's likely that those companies developing will still, as Dan mentioned, have the same issues that their contemporaries that are developing more traditional or small molecule antibiotics have faced. These new therapeutic areas are similarly risky and costly, perhaps even more so in some ways. As Dan mentioned, a lot of questions about how these non-traditional products should be tested for safety and efficacy, how they could be used appropriately in the clinical setting, how are they going to be monitored by antimicrobial stewardship committees there. So if we argue that part of the basis for the lack of innovation may be due to some of the risk associated with discovery, development, and even commercialization, then investors still may have a low appetite to fund these innovative programs much farther, and if the lack of commercial success continues to be the norm. So I think especially some of the first of these coming out of the gates, if they don't get approved or if they are commercially not successful, it would set a precedent even for these to not be that promising. So still significant questions remain and gaps in funding and partnerships and efficient use of capital for AMR drug development. So I think really the key part of this is Jen coming in and talking about those partnerships. So Jen, I'm going to turn over to you to finish this out, please.
Great. Thanks, Brian. And before I get into the nitty-gritty of my few slides, I wanted to put an additional point on the earlier comments on surveillance. So those of you who have been participating in the WHO high-level dialogue on AMR or any of the US AMR discussions recently, you've likely heard the increased language and weight put on One Health and the role of surveillance in understanding disease transmission and other public health dynamics. In addition to this, data generally being important for public health and the ability to conduct clinical trials and other very pragmatic uses, as Dan described, this data is so important to decision makers at the highest levels. So when we can provide concrete data on resistance, the prevalence of these important bacterial pathogens, and really any other data we can get our hands on, the more likely we are going to be able to get that large-scale movement on some of the access and R&D and market challenge that we have historically faced in this space. So as this slide maps out, and as folks working to develop new antibiotics know, the resources in this therapeutic area are quite limited, and it's critical to ensure that all of the money coming into this space is used as efficiently as possible. So for us within GARDP, that means ensuring our mandate is clear and complementary with other funding that exists, and that we don't unnecessarily duplicate what others might already be doing with their own resources. Given that, our focus is on late-stage clinical development and access of antibiotics. We do have projects and programs in discovery and preclinical stages, but the emphasis of our work is on that late-stage clinical development and access piece. The focus of our spend is on serious bacterial infections, pediatrics, and sexually transmitted infections. And this may sound familiar to those of you who know us, as we have partnerships with ANTIBACTERIAL for a gonorrhea program and Venatorix for serious bacterial infections in pediatrics. And before moving off this slide, I'd like to put in a quick pitch for partnerships like those with ANTIBACTERIAL and Venatorix and beyond that. I always say that one of my frustrations in the antibiotic space is that there are only 8 of us who care about this issue and we all keep talking to each other. So I'd like us as a community to really start thinking a bit bigger about who else we might be able to bring to the table, how we might be able to engage new stakeholders, how we might think about new business models, how we might think about investments in a different way. So please be in touch if you have any ideas about this. So moving on to the next slide, please. And thinking about antibiotics and AMR in the context of global health security and importantly, One Health. So as mentioned earlier, we've seen a lot of discussion recently at the global and national levels about One Health. And we recognize that antibiotics can't be thought of in isolation, but rather need to be considered as part of environmental factors and in animal use and in agriculture and in manufacturing and beyond. And this all requires a huge amount of coordination across the public and the private sectors. We've seen from COVID, not just the potential public health and economic impacts of pandemics, but also how valuable partnerships can be and how we can learn from each other and lean on each other to address the various threats. So as we think about antibiotics and pandemics more broadly, as mentioned before, it's critical to ensure that we're thinking holistically about the antibiotic community and who else we can bring to the table. The government can't do it alone, industry can't do it alone, a not-for-profit like GARDP can't do it alone. And I think there are a couple of things to point out, though, in terms of differences between the COVID response and the AMR threat. So first, as Brian, I think, really clearly and nicely pointed out, there's currently a robust antiviral community and a robust vaccine community. And certainly this is more robust now than it was 24 months ago, but there was a pool of resources to build from. And I think a significant risk that we're facing in the antibiotic community is that with every bankruptcy, we are losing expertise. And as the silent pandemic of AMR and the lack of antibiotic pipeline continue to grow, we are concurrently losing expertise, which only further compounds our vulnerability and puts everyone at risk. So governments should consider this dwindling pool of expertise in their own calculus as they consider their response to the next pandemic. Which brings me to my next slide, please. How can we prevent the next pandemic? I think we could all agree that significant investment in antibiotic R&D is required. This includes funding for organizations like GARDP, which can partner with industry on things like pediatric labeling or the conduct of phase 3 and phase 4 trials, as well as ensure there is portfolio management of critical antibiotics, including generics. Significant investment is also needed in manufacturing, which we are certainly seeing a focus on in the US and in other countries, and also incentives for the antibiotic industry to ensure we have a robust pipeline of antibiotics for today as well as we need them into the future. And that investment also goes into securing that needed expertise. As we've been mentioning, we can't continue to lose the scientists and chemists who know how to develop antibiotics. And finally, just a reminder that antibiotics aren't important just to address the issue of antibiotic resistance and the associated public health threat from bacterial infections, but also to address other CVRN type threats and other emerging infectious diseases. As we see with COVID, antibiotics are a critical component in the response to any of these threats. And some of you who are old enough to remember, the original antibiotic programs were established at BARDA primarily to either address biological threats or to manage secondary bacterial infections arising from threats like pandemic flu. So regardless of the lens you use, antibiotics are critical for addressing the next pandemic. So moving on to the next slide. Taking a moment to think about what is really needed to support the global antibiotic community. At GARDP, we believe this means the development of a robust pipeline of technologies, and that includes the associated investment and incentives to support that pipeline, establishing solid public-private partnerships, sharing resources as appropriate, which also enables maximizing capital efficiencies, which I spoke a bit about earlier, building a framework to create access, and in the context of antibiotics, that access also needs to be tied to stewardship, supporting the public health infrastructure, and improving disease and case surveillance. And then my last slide. I just wanted to quickly highlight GARDP's commitment to this space. Our objective is to bring 5 new antibiotic treatments to the community by 2025, and this includes both registration of novel innovator drugs as well as label and pediatric expansion for older products. This requires funding, which we are actively out there fundraising for, but also partnerships. As mentioned, we're always interested to talk with folks to discuss how we might be able to work together to achieve our shared goals. So I think that brings us to our last slide.
Great, thanks, Jen. And just some closing thoughts here and kind of key messages from each of these. And of course, there are some lessons learned from COVID-19 that I think can be applied to AMR drug development. Just as in COVID, older drugs and older antibiotics still have a purpose. We still need to support innovative drugs and continue to look for new ways of managing these patients. Infection control and prevention does work. Arguably during COVID, there might have been some, might have been done more from self-preservation motives and not necessarily altruistic, but they do work when they're implemented. But the resources have been drained, and many of these resources for infection control have been consumed by COVID, so they clearly need to be repleted. Surveillance is an essential pillar to AMR drug development. It's important to know that the prevalence of any resistance pattern may change in the region, and especially if there are surges of a pandemic, it could change that very quickly as different antibiotics are being used or overused. And antibiotic decisions for patients, just as in clinical trial development, have to be informed by regional and local epidemiological trends. Addressing AMR has to become a priority. Clearly, AMR companies are there, they are focused, they are dedicated, and just like as in COVID-19, they have demonstrated they do have the ability to rapidly mobilize and in the face of priorities. When there is a single focused purpose, we can find solutions. The pipeline is probably not where most of us would like to see it, but it is not empty, and there are a lot of opportunities moving forward, and there are partners that clearly have a vested interest, as Jen just mentioned, in tackling AMR, and are very excited to work with your companies and healthcare systems and hospitals that are focused on ways to develop new therapies for AMR organisms. So with that, I want to say thank you very much. We're right at the top of the hour. Hopefully many of you have submitted some questions online where the panelists are willing to stay on, and we can answer some of these questions for the next 10 minutes. But if you do have questions and you have to log off, please send them to us and we can follow up with you offline to help answer your questions. With that, I'll turn it over to Carrie, I think, for any questions, if anyone can stay on.
Thanks, Brian, and thank you to the fantastic panelists and presenters today. Thank you also to the audience for the engaging questions that have come in. So I will go ahead and start with the first question, and I think this one will be directed to Jen. So Jen, you've discussed how non-traditional therapies, vaccines, immunotherapies, and even entirely new types of therapies that have never been approved for use in humans are now being considered to be added to the armamentarium of AMR drugs. How do you feel investors, incentive programs, and policies are considering these? And are they thought to be too high risk for investment or partnerships, or are those programs really being considered?
Thanks for that question. I'll start with my GARDP hat on first, and I would say that first and foremost, our focus at GARDP is on target product profile and access. So more than the class or the novelty of the drug, we're really looking at public health utility, deployability of that asset, how it can be used, how it's filling a public health gap, and ensuring that wherever our investments and our partnerships are made, we're really looking at it through that public health lens first. So that's my GARDP hat. I would say, having previously spent a lot of time working with industry partners, I would say yes, I think generally that these technologies are often very high risk for investment, and I would say that we can certainly look at where some of the investments have been placed previously. That, you know, obviously the market is a challenge in itself, and then when you couple that to a technology challenge, that becomes a risky investment for a lot of investors. So hopefully, as we see the pipeline expand with these non-traditional therapies, with these immunotherapies, with vaccines, and with the added lens of something like COVID, where people start to appreciate really the threat of infectious disease, that we start to see a bit more risk taking in this space, because I would imagine that a lot of us really recognize how useful some of these novel therapies could be, or novel approaches could be, in addressing the resistance threat. So those are my thoughts on that question.
And just to add to around, you know, maybe more just the importance of these, you know, innovative or non-traditionals. When we, you know, many of us are kind of galvanized, if you will, thinking, you know, here's the newest enzyme that's been identified, and here's the newest you know, beta-lactam, beta-lactamase inhibitor combination for that particular new enzyme. But I think what is interesting with some of these newer therapies, it acknowledges the importance of older antibiotics, and just like we've talked about during this call, that they still have utilization. And if you can consider that we have great antibiotics, they aren't necessarily living up to the need that I think any of us think that we have with AMR, or perhaps in the future. If you start to pair those now with these therapies, and many of them are being developed or considered, at least in terms of adjunctive therapies to this, it's a great way now to bring life back to old antibiotics. I'm sure, you know, some of these phage therapies, and as they evolve and newer generations come on, and our comfort level with them increases, you know, perhaps that might change. But now I think in terms of some of the study designs, at least that we see, in terms of how they're using in an adjunctive way, I think is exciting, because you were able to use antibiotics that have been around forever, and maybe we think need to be shelved, could be coming back out because they have a new partner to work alongside them.
That's great. Thank you both for that insightful answer. For this next question, we'll direct it to Dan. What challenges will the new, unique anti-infective product strategies, such as non-small molecule, present to the diagnostic industry responsible for developing tests to detect emerging resistance?
That is a very good question. I don't want to sound flippant, but it's too early to tell. It will. They're so steeped in science that one question is whether or not they will be empirically used, and whether or not you need to profile microorganisms against these products before you use these products. We're the paradigm of small molecules. Yeah, the clinical lab does antimicrobial susceptibility testing, then the physicians look through it and see which drug seems to be best. And I don't know that paradigm is going to hold up with some of these other products. You can get Brian's perspective from a physician, but they're so scientifically and technically complicated that even if there was a diagnostic assay, say for a phage therapeutic, how it would ever be developed so that it could be user-friendly enough for medical technologists and hospitals across the globe to use them seems quite unlikely to me. So I think a lot of these products might just end up having to be used empirically when you know you have a drug, a small molecule resistant organism, then you take one of these products off the shelf. I don't think use of these products, I don't foresee it yet anyway, that the use of these products would be dictated based on clinical microbiology laboratory reporting results on a patient by patient basis.
And you know, I would say the practicality of this, we'll have to see it play out. I think from a clinical trial perspective, you know, it's always we have a study that we're looking to run, and let's say that there is a phage that is active against one specific biotype of Klebsiella. You know, I am aware that companies are trying to develop companion diagnostics, you know, lateral flow assays or what have you to help inform that study, such that patients coming in who have the infection with that specific biotype or that specific organism would be the ones that would be eligible. So, you know, we'll just see how the technology evolves over time and how those studies move forward with that knowledge. If that is the case, you would imagine now that those companion diagnostics would be required, you know, and be part of the label for an approved drug with that. You know, we talk about personalized medicine outside of infectious disease a lot of times, and you know, is this just maybe it's hard to get our minds around in terms of how we're currently practicing and currently running clinical trials, but I think that is a piece of personalized medicine when it comes to infectious disease that could have a very important place. And you know, if it is successful, which again I think we're a little far removed from that fully being implemented, then it does, you know, kind of allay some of the concerns that we have with off-target damage with some of our broad-spectrum antibiotics by using more, you know, precision medicine within infectious disease. It's a fantastic question, and I think it is a topic probably for another discussion at another time.
Yeah, understood. Thank you both. And maybe just time for one more. So Jen, we'll start this out, but I'm sure the others may want to chime in. So we have threat lists, whether from CDC or the WHO, and developers are encouraged really to develop drugs to address these pathogens. But why are we developing drugs which target bugs and not a disease? Is this inadvertently leading to a lot of undifferentiated products?
That's an interesting question. I don't actually have an answer to this. I do think it's probably another one of those questions that deserves some broader comment and introspection. We recognize that the bugs that are on these lists are critical. We need to be thinking about them, but it is interesting, having worked a bit in other therapeutic areas, that the target product profile seems to be the first thing that people pay attention to, whereas often with the antibiotic community, you know, we use these reference lists as kind of as the benchmark of where we begin, and I'm just not sure how that paradigm was created. And it would be interesting to understand, you know, if those lists are inadvertently leading to these undifferentiated products, or how we can ensure that rather than developing drugs to meet these lists, we can think about it in a more integrated fashion to really think about what that public health need is, and fold it into that broader target product profile.
And I think it's back to some of the comments you made earlier too, Jen, is if, you know, there's fear from investors, fear in companies in developing this, and where's the incentive to develop innovative drugs if drugs that are more traditional, you know, are having a hard time finding investors, somebody really pushing the envelope more, how are you going to get an investor there? And I think it just takes that initial leap of faith, if you will, from partners, from investors, from companies to move those drugs forward. And hopefully we have more than luck on our side, and some very great study designs and great products that will come across the finish line successfully, and not only be approved, but show themselves to be commercially viable in some manner that will stand as a precedent moving forward for new innovation. It just, success breeds success. So, maybe I'm overly optimistic, but I fully anticipate that we're going to see that happen. And I think that issue of, lack of differentiation becomes a little less of an issue.
That's great. Thank you so much. And that will conclude our Q&A session. So Sydney, I'll hand it back to you.
Thank you very much all for those answers. And as you said, yes, we've reached the end of the Q&A portion of this webinar, but if we couldn't attend to your questions, the team at MedPace, IHMA, and GARDP may follow up with you. Or if you have any further questions, you can direct them to the email addresses on your screen. Thank you so much, everyone, for participating in today's webinar. You'll be receiving a follow-up from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. Now, please join us in thanking our speakers, Brian Murphy, Jennifer Schneider, and Daniel Stamm. We hope you found this webinar informative, and have a great day, everyone.